So I'm going to be making a series of videos for some common skills that we need to know for a research lab, especially as it pertains to molecular and biology situations. So any of your biochemistry or molecular biology courses. And the first one that we are going to be talking about this week is standard curves. So let's start with talking about what exactly are standard curves. Standard curves are a type of graph that we use many times in our research labs to find quantitative information about our samples. They usually represent a relationship between two different quantities, whether it is the amount of our chemical or amount of our extracted maybe protein or DNA, and some type of dependent variable like absorbance or another situation like that. And in this case, we obviously always need to have some information about some known samples, right? So we need to create the standard curve. So to create the standard curves, we use many samples, multiple samples where we know the amount of that substance that is present. We create that relationship by looking at the absorbance or looking at the optical density, and we graph it, and using interpolation of that, using those samples interpolated on a graph, we can look for that relationship and see what it is, create an equation, and then use it to find our unknowns. So how will exactly we be using standard curves? Well, it says today in the lab that we are going to be talking about for this week, but also in general in this course for specifically our molecular biology as well as the biochemistry courses. For most of the labs, we are going to be using it either to look at the concentration of our diluted chemical that we are plotting. It could be a protein, it could be just a chemical that we are looking at, maybe some dye and its dilutions, or to determine actual concentration of experimental samples, which again are many times going to be some type of protein samples. Our knowns are going to be, if we are looking at a protein sample, obviously then we need a standard curve prepared with purified protein sample. If we are looking at concentration of a different thing, not protein, but maybe a chemical, then we would need our known to be standards that are dilutions of that particular substance. But in our case, if we are going to be looking at protein samples, we are going to create our knowns. Our standards will be prepared using a purified protein. In our case, we are going to use bovine serum albumin with known concentrations. So we are going to take our stock bovine serum albumin that I provide you. You will prepare a set of dilutions and use that to create your standard curve. Now let's say, I don't have any bovine serum albumin, I lose it, or it's on back order and we don't have any. Well, that doesn't mean that we cannot create a standard curve for our protein samples. We can still do it. And you can think about what else can we use instead. So again, remember, to create a good standard curve, or to create a standard curve, you need a known of that purified sample of that type that you're looking for, whether it's a chemical or a substance. And then you need to prepare a set of samples with known amounts present in each one of them, usually done by dilutions of your stock solution. So if we don't have purified bovine serum albumin, which is a purified protein essentially, we can use some other protein, right? As long as it's a pure protein sample of some sort. It could be caspase, it could be albumin, it could be actin, it could be anything. It could be Krebs, it could be IgG. Does not matter what it is, as long as it's pure protein sample and we know the starting concentration of that protein, so that we can generate a set of dilutions from it with known amounts of proteins in each one of them. We can utilize it. If you don't have any one of those, you could still even think about a situation where you can go to GNC and get pure whey protein. As long as it doesn't have any additives, you can utilize it to create your standard curve. Now, once you've created those dilutions, you can utilize many different types of methods to look for amounts of proteins in there and create relationships. Each one of those methods are going to give you different types of relationships, right? Not everyone is going to be purely a linear standard curve. So for pure protein samples, without using any of these other, additions, you can still look at their absorbance, right, at 280 nanometer wavelength, which is going to look at the amino acid content in your sample and use that to create a relationship as well. However, many times what people use instead are one of the common colorimetric assays available in the market. These colorimetric assays are going to convert into, are going to produce a color change when they interact with your protein samples, and that color change is what's measured with the spectrophotometry. And obviously, when we look at that, increasing absorbance is going to lead to increasing amount of protein, and that's your relationship. Now, there are three methods that are used very commonly. Two of them are classical methods. They're the historical methods that have been used in the past. One of them is Biuret, and the other one is Lowry. Both of these have been used for a long time. They both depend on copper ions binding to the N-terminal of your proteins and leading to a color change. Biuret method works under alkaline conditions, and while it is, it's very linear, it works very well in some conditions, it also has a lot of backdraws. It can give this white precipitate, which can obviously mess up your absorbances, so you don't get very good relationship. It also requires the samples to be boiled, and, it's cumbersome, so it's not going to be the best one if you want to protect your protein samples at the end of the day. And it's not going to be very good at that quantification. Lowry method doesn't involve as many, boiling and other types of things. It doesn't get any of that precipitate, but it still has the issue that it actually takes about 2 hours to, complete from beginning to end. So that's going to be a really long time. It's very linear. It gives you very good range of proteins that can be looked at in that linear range. So that's a good positive, but 2 hours is a long time, especially if you want to protect your proteins and want to analyze them immediately afterwards. There is a newer method that is kind of a new way of using the copper ions called the BCA assay, and that only takes about 30 minutes, and that's very quantitative. It's within the time range, so that's another way that we can look at our stuff. A lot of times in research labs, that's the method that we use. However, sometimes we really need something quick and simple and be able to move on to the next step very, very quickly. Maybe because we want to protect the enzymatic activity of our protein, or we are in a time crunch and need to go to the next step quick. So for those timings, is another method called Comasi blue dye binding assay. It's also known as the Bradford method. In this particular assay, the dye actually binds to protein under the acidic conditions, and it causes a color change that can be looked at 595 nanometer. This is an immediate reaction, so you add the method, add the substance, you mix it up, and you're ready to look at it right away. Because it is so quick, it has to be measured immediately, or it will saturate and you will run out of that linear range very quickly. So sometimes you may not get a beautiful, simple linear curve to look at. You may have to actually do a logarithmic or polynomial equation instead. However, again, the biggest, advantage of this is that it's very quick. You can get those numbers very quick. One of the disadvantages of this method is that, obviously one disadvantage is that because it is so quick, you can run out of the linear range and saturate. But a second disadvantage of this method is that this particular dye can be, it's not always going to be able to be used with your particular lysis buffer. Some lysis buffers will give you just a saturated response immediately. So in those cases, it's not compatible with those lysis buffers and you cannot utilize it. But if you have a kind of gentle lysis buffer that is compatible with Comasi blue dye binding assay, then it's a very good assay to use. We're going to be using this assay in this class because we want to see all those different ways that your standard curve can show up, and we want to be able to understand how to choose the correct one under any specific conditions. So just to remind you again, the Bradford assay is based on the Comasi Brilliant blue dye binding with your proteins. under a in normal conditions, it is going to be just when it's present present in its positively charged cationic stage, and it's kind of this red ugly brown color in their normal anionic conditions. When it binds to your proteins, those are stabilized, and it changes into its negative anionic form, which causes it to convert into the blue color, which is what we look at. Now, it is more biased towards the basic letters because, again, it's working under the acidic conditions and it's normally positively charged. So it's more likely to bind to arginines, lysines, and histidines in your protein samples. Most, different proteins are going to have different ratios of those particular amino acids. So you can imagine, some proteins are going to be obviously more represented within it than others. So it's not necessarily that it's looking at all the proteins at the same level. There's another way that it also binds to your proteins, and that's because it has this, ring-like structure. It can kind of hang on to your ring R groups in your amino acids. So tryptophan, tyrosine, and phenylalanines, which have those ring structures, would also kind of bind to it as well at some level. So you will have some of those represented in there as well. Now, for the first lab, we are going to practice creating a protein standard curve given a purified protein sample, a standard protein, which is our bovine serum albumin, and we are going to use it to practice graphing our standard curve and then choosing what would be the most appropriate function to look at, what would be the most appropriate equation to look at given the way our absorbances turn out. The purified protein, your first tube is going to contain just plain water, which is our diluent. It's milky water in this case. That's going to be our blank. That's going to be looked at as our background noise from our samples when there's no protein present. And our last sample, number 9, is going to be just your pure stock BSA, which is 2 milligrams per milliliter in our case. So that's what we're going to be using for that. You're going to work with your partner to create the protein standard curve, because you're not just going to make one single set of dilutions. You're going to be doing a replicate of 3. So your first, 3 rows on your plates, those are going to be your BSA standard dilutions, going from 0 to 9, right? So these 9 samples. I'm not giving you the concentrations in milligrams per mil of those particular dilutions. You need to calculate those using the C1V1 equals C2V2, my favorite formula. We'll go over that in class again, just to make sure everyone knows how to do those. And then I'm also going to be providing you with an extracted protein, which is kind of like a known sample for me. I know how much protein should be present in there, the concentration of protein in that sample. However, you are going to be using that as your unknown, and you will also be plating that in triplicate. Your first well, again, is going to be just your diluent, the water sample. That's your background noise again. And then your second sample is going to be a 1/2 diluted sample of this extracted protein that I provide you. And your last column is your undiluted pure protein that I provide you. You are going to take the standard curve that you create from your BSA standard dilutions, and you're going to utilize the equations from there to find the concentration of the unknown sample. And that's going to be your first exercise. Now, why do we need exactly to create this 1/2 dilution? Why can't I just plate my, fully concentrated undiluted sample? Well, the reason for that is that any standard curve that you create is going to be best utilized for the concentrations that it is made for. So if your dilutions that you prepare from your BSA go from 0.2 to 2 mg per mil, then that's the range of that standard curve. Your standard curve is best utilized to quantify any protein sample that falls within that range. So if I give you an unknown sample that's 3 mg per mil, or 0.125 mg per mil, that particular standard curve that you would have made is going to not be suitable for that sample, because your unknown is falling outside the range of those standards. Now, if it's too low, well, you're kind of in a mess. You have to redo a standard curve that contains those lower values. But if you have too high a sample, that was something that you could have prevented all the rework for by simply creating some dilutions. So to prevent the shot in the dark technique, what we do is we create this dilution of 1/2 so that just in case our fully concentrated sample is too high, our 1/2 will fit the curve and we should be able to get a proper quantification from the same standard that we've already created. In this case, we just calculate back to determine the amount of protein in our original sample at the end of the day by taking the dilution factor into account. So if for us, because we are creating a 1/2 dilution, we'll just multiply the answer by 2 so that we can get the actual concentration in our undiluted sample. So as we talked about it, that our standard curves using the BSA with the Kumasi Bradford dye may not give you just a simple linear trend line. The way we're going to be looking at this is, first of all, to create that scatter plot. To create the scatter plot, you're going to average the absorbances for each of your standards, those 3 replicates that you got. You're going to subtract the average blank value that you have from the 3 blanks, the first well. And then you're going to subtract that value, remove that value from all your samples to get the blank corrected absorbance. You're going to then generate an XY scatter plot in which your BSA concentration, which is your independent variable, is on the x-axis, and the absorbance at 595, which is your dependent variable, on the y-axis. Now, you can use the 0 in there or not. However, in our case, we are going to omit the 0 value. So you will not be plotting the 0, 0 point. You're only going to be plotting your actual dilution. So, well, 2 through 9 is what you should be plotting. Once you have plotted those samples in your Excel graph as a scatter plot, you will then add the trend line and you will examine the various trend line options to see which one fits the best. Now, here you see 2 common trend functions that might be possible, right? A linear or a polynomial, which you would be able to look at and then examine the equation and the regression value to see which one is the best. Once you decide which one is the best, you are going to be then taking the absorbance of your blank corrected unknown sample into that equation. And again, your absorbance is the y, so you will plug that absorbance into the y in that equation and solve for the x to examine, to get your quantification of the unknown protein. In Excel, there are 3 options suggested for determining the trend line. You have your simple linear equation, which is obviously the simplest to find, to solve at the end of the day. So, if you can get your samples to be in that linear range, that's great, because then it's easy calculations, mx plus b, simple. Or you can have a logarithmic or a second degree polynomial, which obviously would then require you to use the quadratic formula and be a little bit more complex. How do you determine which one best fits the data is going to be dependent on your absorbances, how quickly you were able to, how accurately you created your dilutions, how quickly you were able to add the Bradford dye and read that on the spectrophotometer. And the best way to examine which one is best fitting your graph would be to look at the regression value. So here is an example data set where you can see, obviously every single substance you examine, anytime you create a series of dilutions from super low to super high, you're going to see that there are going to be some parts of your equations that are, of your scatter plot, that are going to be outside the linear range, that do not follow Beer's law, either because the concentration of your substance is too low or too high for the instrument that you're working with. If you have very, very low concentration of protein or the substance you're examining, maybe it's not going to be able to give you that relationship accurately. Similarly, in every single case, there's going to come a concentration where no matter how much more you add, it's already super saturated. It's going to kind of plateau out. So the sweet spot is obviously this linear range, but your samples may be, some of them may not be in that linear range, which will force you to use one of your other trend functions. The way that you're going to choose which one fits the best is going to be simply by looking at the regression value for now. So this is what you're looking at. In any of these lines, the closer the regression value is to 1, the better fit it is. So between these three, in our case, in this particular example data, the second order polynomial trend line seems to fit the curve the best. Now you have the option to omit some of these points, up to 2 points, and see if you can get it to fit into a linear trend line. That would be great because that will make your calculations easier. If not, you know which one to use because that one is the one that best fits the curve that you get. So in this case, I would be using the second order polynomial trend line unless I remove some of these points and see if it can fit one of the other functions better. So hope that helps and gets you ready for this first exercise. We're going to be utilizing this particular work again and again all throughout the semester with each of the modules because each module will require us to extract some protein, quantify it, and then examine it in some way, whether it is through page, running it on a gel, running it using an antibody, by Western, or by quantifying it through ELISA. So it is a very important skill that we want to make sure everyone gets it in this first week.